right. We've covered prokaryotic cell structure. We talked about differences between prokaryotes and eukaryotes. Remember, this class we are focusing on those bacteria. We need to talk about how those bacteria grow. We need to talk about how we're growing them in the lab and how we're doing things in the lab. You know, a question I often get is, well, where is lab content on the exams? Lab and lecture go hand in hand. The stuff we're doing in the lab is stuff we are talking about in the lecture. So all of that information is useful. All of that's coming back to some of this. So we're actually going to spend quite a few labs going through some of these components on how do we grow microbes and what are we looking at. Here's a bunch of different agar plates. What do you notice about these? What would you say?
Colorful.
They are colorful. What parts are colorful?
All of them.
All of them. The augers, different colors.
Specimens.
The organisms in some cases are different colors, okay? All of this are different types of growth media that we could use to grow organisms on in the lab. One thing that I want to stress is that not all organisms grow on every type of media. Just because you put something on a plate doesn't mean it's going to be happy there, doesn't mean it's going to grow there. The other thing is that as we vary the nutritional content of the media itself, we can entice those organisms to grow and to then change color so that we can tell them apart. So we can actually encourage some of these color changes to help us understand what's going on, what's growing on the plate, what do you have, what do you not have. Later on in class, towards the end of the semester, you are going to swab your nose, you're going to swab your mouth, you're going to swab a different skin area of your choice. We're going to put it on different types of media. One type of media that we put it on is called mannitol salt agar. That mannitol salt only grows species of Staphylococcus. So all of the staph you've got from that area you swab are going to grow on that particular type of media. But if the species that you are culturing, if one of the organisms that you have is actually Staphylococcus aureus, that organism is going to turn yellow. So we're going to know just from growth on that plate, yes, you have Staphylococcus in your nose, in your mouth, something like that. If your plate also turns yellow, we know not only do you have staph, but the staph that you have is Staphylococcus aureus, all because of the nutrients and the components in the media. Okay? So what we choose to grow organisms on is really specific, and we can really manipulate that. These organisms that we are working with, they are prokaryotes. Think back to your cellular division. You learned about mitosis, okay, a very long time ago. You learned about the fact that, hey, as the cell needs to divide, it needs to copy its DNA, okay? And then that cell gets larger. Those structures are going to, alignment of the chromosomes, pulling the chromosomes to different parts of the cell. You went through all of those different phases of mitosis before. Good or bad, these are prokaryotes. They don't have a nucleus. They're fairly simplified. And every daughter cell is identical to the parent cell. So there's no rearrangement of chromosomes like what you see in mitosis. Therefore, this is a simplified process. These guys are using binary fission to divide. The process of binary fission is relatively simple. Okay, so what happens first is that DNA actually attaches to that cytoplasmic membrane, and it's going to multiply. So that's no different than what you would see in a eukaryotic cell. You've got to copy your DNA so that each daughter cell is going to have one copy. Then it actually enlarges a bit, so it's got enough cytoplasm, enough ribosomes, all the internal components. And then what happens here is that it actually forms a cross wall, and it splits off, partitioning the DNA into two cells and making two separate daughter cells, okay? Through this process, you can also see how these cells might stick together, like what we talked about last Wednesday, where we said these cells typically group together and stay along the plane after they've gone through cell division. In this process of binary fission, everything is identical, okay? The cell population is going to double every time division happens. So if you had one cell and it divides, you've got two. Two cells divide to make 4 cells. 4 cells divide to make 8. So the cell growth is said to be exponential. And then what I have written here is that exponential growth has important health consequences. What this means is that a small number of cells now can be millions of cells in just a very short amount of time. E. coli, for example, can multiply depending on the species, can multiply in as little time as 15 to 20 minutes, okay? That's it, that's all it takes. You never end up with just one E. coli cell that you've ingested or that's on your hands or something like that. You end up with hundreds. And those hundreds start multiplying every 15 to 20 minutes, and suddenly that cell population gets out of control very quickly. Food poisoning caused by E. coli gets you very sick, typically within just a matter of hours, because the cells multiply very fast when they end up in the wrong place at the wrong time, okay? When you don't see growth on something today, but you see it tomorrow, it's a result of cell division. In other words, you know, you got bread on your counter, and tomorrow it looks moldy, but, you know, today it didn't. Well, chances are the mold was on there today. It just wasn't a number high enough that you could actually see it yet, okay? So what we have is what's known as the doubling time or the generation time. It's the amount of time it takes for that population to double. Again, for E. coli, that's as little as 20 minutes, 15 minutes, depending on the species. Most species that are going to cause human infection are going to multiply fairly rapidly, okay? That doubling time is really going to vary for every type of species on a plate. When we looked at your hand washing plates, when we looked at your environmental culture swabs last Wednesday, we said a way that you can tell two species apart is by the size of the colony, okay? Some of your colonies were really small and other colonies on your plate were really big. That's got to do with the generation time, the doubling time. The faster that doubling time is, the larger the colonies are going to get. The slower, the longer it takes for that doubling time to occur, those colonies tend to stay smaller on the plate. So you can tell species apart by how fast they double. We can see that, visually speaking, on a plate by looking at the colony size. We can also calculate how many cells are in a population at any given point in time. We can calculate back to the original number. We can calculate out to the final number. So the bacterial growth calculation is listed here. So it's n sub t is equal to n sub o times 2 to the nth power. Please note, it's not 2 times n. It's 2 to the nth power, okay? So n sub t is the number of cells in a population at a given time. So think of that as your final population number. N sub O is the original number of cells in the population, and little n is the number of divisions. So if you have 800 cells in a population, after 3 generations, how many cells did you start with? Okay? All you got to do is plug in the numbers according to the calculation. So we have 800 is our final number. We are trying to solve for n sub O. 3 is your number of generations. 2 to the 3rd power is 8. Yeah. 800 equals n sub O times 8. See what I did with the math here? I made this really nice and easy. Okay? 800 divided by 8 gives us, so we started with 100 cells. We allowed 3 generations to occur. After just 3 generations, we went from 100 cells to 800 cells, okay? If we had 10 cells in an original population and we allowed 12 divisions to occur, how many cells do we have in our final population? 2 to the 12 is 4,096 times that 10 original number. Our final population number is 40,960, okay? In the event there is a 20-minute generation time or doubling time, okay? This process only takes 4 hours to occur. So in 4 hours, if you started with 10 cells and it multiplies every 20 minutes, in just 4 hours you can go from 10 cells 40,960 cells. The number grows really quickly. If I said it takes 4 hours for those 12 generations to occur, what's its doubling time? You could literally just convert that 4 hours into minutes, okay? 240 minutes divided by 12. Okay? We're making sense in how to use this calculation? The biggest way people go wrong is you don't remember that the little n is an exponent. You think you're just multiplying. You're going to end up with a drastically lower number than what you should if you just multiply. Okay? Understanding how many cells are in a population at any point in time is really important because it helps you to understand, if it's a patient sample, it helps you to understand, okay, how fast is that organism growing within that patient. If you are taking a culture from your patient and you're trying to say, okay, how long do I need to let this grow for me to help understand if it's positive for cells or not positive for cells? Well, you say, okay, depending on what could be in that sample, I know what my generation time is. I know how long it's going to take me to detect it. You can work back, okay? So understanding how many cells are in a population can be really important from a medical standpoint. It's also really important from a research standpoint. If you are studying how an organism grows and divides or how to hit it with antibiotics, how many antibiotics, what the dosage is of that antibiotic, you need to understand how many cells are in that population. If you have an organism that's making a product you want to purify out, you need to understand how fast it's growing. For example, bacteria are used to make human insulin. They only make human insulin when they're in a certain growth range. Too many cells, they exhaust the nutrients, they're not making insulin anymore. That product that you want to drive is not being produced. By understanding how many cells are in that population or in your culture, you can make sure you've got the right number, you can make sure you also have the right nutrients, and you can make sure it's always producing the product that you want to have occur or released. So anytime cells are growing in kind of a fixed scenario, they are going to undergo a very specific growth curve. When you put them into a new environment, when you allow them to grow, when you don't add any new addition of nutrients, this is the growth curve that they are going to follow. Okay? And what this looks like, you've got 5 main phases. What you can see here is you've got time, hours, and then later on this is going to prolong into days and years. So the 5 main phases are the lag phase, log phase, which is also called the exponential phase, stationary phase, death phase, and the phase of prolonged decline. Typically we recognize through the first 4 phases, and this stage of prolonged decline is kind of what's left over, okay, if you don't manage to kill everything off. So the lag phase that you can see here, notice this is cell number in the log scale on the y-axis. So that lag phase is level, stagnant. In all of these, in this case, the cells are still alive, but the cells are kind of gearing up. They're tooling up for division. They're getting to know what their environment and what they're doing. Okay, this is kind of like you start a new job. When you start a new job, you're never productive the very first day. You're like, let me get to know the people I work with. What is my job? How do I do it? There's always kind of a lag time where you're not really doing much. That's what's happening in a culture when you introduce organisms. Then once they really realize, hey, wait a minute, everything is good and fine, I'm going to start to divide, they end up in the log or the exponential phase. Here is where they're dividing at that optimum rate, so growth is going to be exponential. What happens here is that exponential growth is not capable of being maintained, okay? Because these organisms are using up all of the nutrients here, they're going to run out of nutrients, and then they're going to start to be like, wait a minute, we can't divide as fast when we don't have as many nutrients. And from there, they end up in the stationary phase. The stationary phase is also flat. It's flat here because the growth is equal to the death. The cells that are dying off are providing just enough nutrients to maintain the cells that are left and still dividing. But the growth is equal to the death in this case. In the death phase, you're no longer able to sustain that, and there is more cells dying off than are actually growing or dividing. And then finally, they hit that phase of prolonged decline, and this can basically keep going down, 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 down. How fast they hit this, so death is also exponential, but how fast they hit this and how fast this line keeps going down is going to be dependent on a lot of different things in the culture at any given point in time. Okay? You have to understand the growth curve. When we grow organisms in class, we try to maintain them in the log or the exponential phase. That's when they're happy. That's when they're healthy. That's where they're dividing nicely. That's where their cell wall components are all nicely formed and fully functioning. If they're going to produce metabolites, it's going to happen in the log phase or the exponential phase. Once cells hit the stationary phase and then into the death phase, they're not happy anymore. So doing biochemical tests, staining them, all of those things they don't really like to cooperate with anymore when they're not in an optimal functioning state. All this is just a little bit more explanation. So again, lag phase, number of cells isn't increasing, but they're still alive. They're metabolically active. That log phase, they're exponentially growing. They're going to be producing metabolites. If they are making antibiotics, so organisms can naturally produce antibiotics. Those that are doing so are doing that in that late log phase. Okay. Producing metabolites is really an important component. When you're doing research, it's an important component as well. They, again, trying to keep track of that cell number, how fast they're going through binary fission and where they're at in the growth rate becomes really important in studying bacteria because you need them to be happy. You need them to be healthy. You need to have them be performing optimally at any one point in time. That stationary phase is when we've exhausted the nutrients and the growth is equal to the death. The death phase, the total viable number of cells declines and death kind of keeps going from there. Now, this is when you've got no new input of nutrients, not a lot of extra stuff coming back. If you do have nutrients coming in, it depends on how fast they're dividing as to how well this is going to be sustained or maintained by the population. Think about it this way. That bacterial growth curve is important if you want to take an antibiotic. Okay? Let's say you go to the doctor, you've got an infection. They prescribe an antibiotic. They say to you, you've got to take this for 10 days. How long do you take that antibiotic?
10 days. You're supposed to take it for 10 days.
Always take it the 10 days, okay? Why?
Most people just, they feel better and stop taking it.
Worse. And it hasn't gotten it. It hasn't killed it all. It's just stagnant.
It hasn't killed it all. It's just stagnant. You feel better. Sure you do. Because that antibiotic is like, okay, wait a minute. We're starting to work. We're starting to kill them off. I'm feeling better. And so people go, oh, I'm not taking it anymore. Guess what? You haven't killed off enough of these cells. That 10 days is to get you way over here so that those cells are next to little to nothing, and they're hopefully not coming back. I talked to someone last week, and she had a whole bunch of stuff, and then she was just like, well, yeah, we never take the antibiotics for the full round. I always save something because I'm going to get sick with it again. You're going to get sick with it again because you didn't take all the drugs the first time. And I was like, oh, here we go. Here it is. I was like, you are a grown-ass woman. What is wrong with you? Let's talk about this. And she was just like, well, no, why would I do that? I felt better. When I feel better, I stop taking it. That is the wrong thing to do. Because this is going to, it's going to start wiping out these bacteria. You start to feel better. But you have an infection. You have signs and symptoms because you have lots of these organisms going through your system. And your immune system needs some help at wiping it out. So you need to get it way down here. Your immune system, your normal flora, other parts of your body will all help the rest of this out. But you're taking the antibiotics for a reason. It's all coming back to this.
When they say that you can't drink alcohol on antibiotics, is it because it weakens the effect of them? Or what's the point of it? I'm just curious.
Just curious. Okay.
It's just a suggestion.
Yeah. Don't. The question was, should you not drink alcohol with your antibiotics, and what's it doing when you get into that? Yes. In theory, the alcohol is affecting your immune system. It can make certain parts of your body work overtime, depending on how much you drink, and how fast you're metabolizing that and excreting it is going to affect the overall half-life of the drug and how long it's active in your system.
It doesn't necessarily affect the efficacy of the drug, though.
No, it's not necessarily it's not like it's binding to the drug. And
all I'm hearing is it's okay to drink iron.
It's.
She's not
This is what gets me into trouble.
No, this
okay?
was that. I'm just curious because I drank on every antibiotic in college, so I'm just curious.
Oh my lord! Right.
I don't do it now.
Part of it has to do with how much you're drinking and how fast you're then excreting everything from your system. because your body then works overtime to get rid of the alcohol. You're purifying all of the antibiotics and everything out with it too.
Oh, gotcha. Okay.
If you're within, if you're moderation, you should be fine.
No heavy drinking when
No
you're
heavy
sick.
drinking when you're, yeah, that would be good.
Yeah,
It's
that
also
was-
bad for
that's
your immune
probably also
system
why I
and
was sick
everything
all the
else.
time.
Yeah,
Take it off. Take it off.
that's fine. All right. Again, you want to get that population in that phase of prolonged decline. Nearly 99% of all cells are dead. Then you know everything is gone. Okay. This may last months or years. It depends on what the culture is. Depends on the scenario. When this is in your body, you can have it where that number goes really low, and then that number could come high later on. You are someone who gets chronic infections. It is possible that you reduce the numbers, and then they slowly grow back over time. You're just fully not getting rid of them. People who get chronic urinary tract infections, chronic sinus infections, chronic ear infections, in those cases, most of the time you're reducing the number of organisms, but it's not taking it down to a level that's low enough where it's not going to come back later on. Okay, so it really depends when this is in your body. It depends on the site. It depends on what that organism is. When this happens in a culture, it really just depends on the exact conditions that you've got. Okay, when we grow things in the lab, we can have things that sit in the fridge for a year or more. Okay, you can get them out and you can revive them. They might not be great, but there are typically some cells within that population that are still going to be fine. Okay. So that's controlling everything. That's doing something in a set environment. In nature, on the other hand, you have a lot of extra conditions that are all present, that are all contributing to growth of that organism. Please keep in mind there's a number of things that are going on here. These bacterial cells, these prokaryotes, do have the ability to sense that environment. They understand how it's changing, and they can produce compounds and structures that allow them to survive inside of a changing environment. They also rely on other organisms in the area to help them survive. Okay? An example, so biofilms and slime layers. Plaque on your teeth is basically a biofilm. Okay? It relies on a set number of organisms to come in. They attach to your teeth first. They basically glue on there like cement. And as other organisms go by in what you eat and drink, fingers in your mouth, all that fun kind of stuff on a regular basis, they then attach to the other bacteria that are stuck onto your teeth, and it builds on this really awesome layer that you then scrub off when you go to brush your teeth. Okay? But that biofilm is cool because those cells in the biofilm are actually feeding off one another. I have this, I'm giving this off as a product, you need it, you can use it as a nutrient, here you go. Hey, you have excess of this, I need some, give it to me. So they're actually really cool at kind of sharing and swapping things, and making sure that everybody in the biofilm survives. So in nature, nature is very much a mixed bag. It's not a pure culture, it's not one organism over here and one organism over here, it's everything on top of one another. Those organisms can either be happy, and they can work together with one another. In some cases, they make associations that poison their neighbors. They just don't get along, okay? This is no different than, you getting along with your family, okay? Some days, everybody is happy and everything is fine, and other days, you just want to wring their neck, okay? Walk away. Block them on Facebook, whatever it happens to be, okay? These bacteria are doing the same thing. They're all on top of one another. They're interacting with each other on a regular basis. So that biofilm, again, I kind of explained it as you're eating and drinking, okay? The technical term is planktonic bacteria. Planktonic means free-floating. So when free-floating bacteria attach to surfaces, they start to grow, they form an initial layer, and then others can grow on top of that. Anytime you've got a biofilm, it's got a really characteristic architecture that allows nutrients to come in, waste out. If you've ever been in a pond, you go walking in a stream, creaking, as I've heard it is called around here, okay? You step on stones, they're like slimy, okay? What is this slime? It's a biofilm, okay? All it is all kinds of bacteria that have made a biofilm, okay? If you ever go into a pool that's not properly treated, the sides will feel all slimy. It's a biofilm, okay? most of the time, that biofilm can actually be purifying in nature, okay? If biofilms can be used to purify your water, believe it or not. So they make these water columns, they build up the biofilm, and certain organisms will purify out different waste products. At the bottom of the column, that biofilm has actually done a lot to get rid of all of these different products that we don't want in our water. So biofilms can be fine. Biofilms can also have organisms in it that cause disease, like plaque on your teeth, okay? You understand, you end up with plaque, that leads to things like cavities, okay? And that gum disease, all right? Not necessarily great, okay? What was that?
Gingivitis.
Gingivitis. See, fun stuff. Organisms that cause gum disease typically are linked to heart problems and other issues, okay? So once there, you've got a biofilm that can be hard to get rid of. If you end up with like a pacemaker put in, organisms can travel and form biofilms on a pacemaker that can lead to all kinds of other complications. So cells within a biofilm can cause disease, depends on what's in the biofilm and where it's at.
So for biofilms, does a lot of it come from like your genetics? Like what type of bacteria is growing?
So what type of bacteria is growing to make up the biofilm depends on that location. So if it's for your teeth, for example, everything that you eat and drink, that's what's bringing in the bacteria.
So how do genetics play a role in that? Because like I know for at least like cavities and stuff, a lot of that relies on like how your parents' genetics are.
So part of that is dependent upon the state of your enamel and how thick your enamel is and how sensitive your
Okay.
enamel is. That part is based on your genetics. You likely have similar habits to your parents, so that then plays a role in it as well. How often are you brushing your teeth? Are you doing a good job? How well do they teach you to brush? That, all of that kind of plays into it. But what happens in a cavity is that the organisms, when they form that biofilm, they basically release acid. The acid sits against your teeth, and that destroys the enamel. Once you get down to a certain extent, it basically drills holes in your teeth, and that leads to a cavity. So how thick or how well that enamel is formed, that's going to depend on, are you more likely or less likely to end up with cavities. The bacteria themselves are going to be completely
This-
external component. We can use biofilms for bioremediation, so cleaning up things like oil spills, okay? Cleaning up toxic waste in the environment, putting certain organisms back, encouraging them to grow on certain surfaces helps to clean up the water, okay? They've done things where they've basically released solid objects into a water column, and those solid objects were colonized with certain bacteria that were meant to help purify and clean out that water, okay? So we can do a lot of good things with biofilms. We can do a lot of, they can cause disease depending on where they're at as well. What I want you to remember is that in nature, organisms are all mixed up. They're not separate, and they're interacting with one another. One way to interact with one another is to create a biofilm where you're sharing nutrients, you're sharing components to basically make sure everybody gets along, okay? So the waste of one organism could be a nutrient for one another. In certain scenarios, these guys can compete for nutrients. They can produce toxic substances to inhibit others as well. If you have siblings, think about it like your sibling, okay? Some days you get along with them, other days you do not, okay? Same thing with bacteria interacting in nature, okay? When we try to grow organisms in a lab, it's really hard to reproduce that exact set of associations. So trying to study how it is you've got some sort of disease can be really complicated, especially if you've got a disease in a location that's got normal flora present, okay? That's got all kinds of other contributing factors. We can't just magically reproduce what's going on in your gut in a petri dish. There's too many other factors that are going on there. So sometimes people want to say, well, they can't figure out what it is I've got. Yeah, that may be complicated if you've got 5 different organisms or more in an area. If you've got conditions of high acid or low acid, what are all of the different extenuating circumstances to lead to that exact disease? It gets complicated. So trying to basically study all of these different diseases in a research lab is not necessarily the easiest thing. We don't just take a swab from somewhere and culture it and immediately figure out what it is you've got and how we treat it. There are a lot of complications that can go into certain things like this. So when we grow cells in the lab, we've done this already. Remember, we streaked them on that little plate, okay? That plate is considered to be a closed system. It's also called a batch system, okay? There's no new input of nutrients on that petri dish, okay? That agar is set. When the agar has no more nutrients, the nutrients are gone, the organisms are dead. So closed systems in a laboratory can be good. They can also be bad if you're trying to maintain it on one particular type of medium, okay? All because you're not adding in no additional nutrients. You're not taking away the waste. But the population of cells in that scenario will increase in a very predictive manner. They will follow the growth curve. When we grow cells in the lab, we control how long they stay in the incubator. We control then when we move them to the cold. Because we only want growth to occur for a set amount of time. We want them to end up into that exponential growth phase. And then we want to move them to somewhere that's going to slow them down because we don't want them to all start dying off on the plate, okay? The longer you let those organisms grow, the longer they're going to then go to exhaust the nutrients and then start dying, okay? You have to control for all of that. In the lab, we're typically going to be growing things in what's known as a pure culture format. What we did when we had just swab your fingers and put them on the plate was a mixed culture. We took everything that was on your fingers, we got them onto the media, and we said, okay, here you go, all right? Commonly when we grow organisms in the lab, however, we're going to create a culture that has only one type of organism growing on that particular plate, okay? We are going to do this using aseptic technique, so we're going to talk next week about sterilizing our loops and how do you make sure something is sterile. When I say in the lab, we talked about this 2 weeks ago, I said, look, hold that plate like it's a clamshell. Make sure that that lid is shielding the surface of the media so that you're not getting cells to drop in over top of it. That's part of aseptic technique. Not spitting on the plate while you are inoculating it is part of aseptic technique. This is no different than why does a physician put on gloves when they come in your room? Or why do they really scrub in, wear a face mask and a gown when they're going in for surgery? All of that is aseptic technique to try and reduce contamination. In class, we're going to grow cells in what's known as culture media. Media is plural, medium is singular. Media can be in multiple forms. We are commonly going to grow things in class on a solid form, which is an agar plate, like what we used a couple weeks ago. But we can also grow them in a liquid as well. So, the question is, how do you know if you want to see something in a colony format, how do you have to grow it? Well, if you're going to be looking at colonies, they need to be growing on a solid surface. Okay? What you're looking at here is a solid agar plate with all kinds of individual colonies growing on it. It's solid because it's had the addition of agar. Agar is actually marine algae agar extract. It liquefies at 95 degrees. We make this like we make Jell-O, okay? You add in the different nutrients the bacteria needs. You add in a set amount of agar. You boil it so that you liquefy everything. It then gets poured into all of those individual petri dishes. And as it cools down, it solidifies and takes the form of the dish, okay? If you've ever made Jell-O, it's the same exact process, okay? But what we do then, we can then have the nutrients in the plate and we can grow the organisms on the surface to see separation of colonies, to help us tell differences in species. It makes a really nice, solid medium. On the other hand, we can use almost any liquid to grow bacteria as long as we have the right nutrients, okay? Liquid is actually better if you want to grow larger numbers of media, or of organisms. Most industrial uses for microbes and getting them to produce products are growing them in a liquid and not on a solid because you can grow them faster, better access to nutrients when you're in a liquid. You can have the same exact nutritional components and have one be a liquid media and one be a solid media. The only difference there is that addition of agar, okay? So when would we want to use liquid media? When we want to grow larger numbers, okay? We want to grow them faster. Liquid is better for those organisms to have equal access to nutrients. Solid media is better if you want to see them in individual colonies, if you think you have more than one species that you want to tell apart. When you're not concerned about how well they're growing, colonies then can be better, okay? When you see this colony on a plate, So the cell started in the very center of that colony, and it's undergone binary fission and mounded on top of itself. The cells at the edge of the colony actually have little competition for nutrients. The cells at the edge of the colony are actually better fit-wise than cells at the center of the colony. So when we are in class and I say, look, let's take a swipe from that colony off the plate, you want to pick it up, you want to put it on another media. You're always picking up from the outside part of the colony, because those are going to be the cells that have best access to nutrients. Those are the cells that are happiest. The cells that are in the center of the colony are all crowded on top of one another. They have less access to nutrients, lots more competition, less access to gas specifically. They're not as happy, right? So there are pros and cons to using each type of media, liquid versus solid, okay? Liquid, good for growing lots of numbers of organisms, but not good if you want to tell them apart. Solid surfaces, much better for telling apart how many species you have there, getting them to grow in colonies, getting additional characteristics, but not so good if what you need are large numbers of cells. Please understand that if these guys, if you see it on a plate, the teeniest, tiniest dot on a plate has at least 1 million cells. So if you can visualize it, it's got over 1 million cells in it. The bigger colonies that you were looking at last week had millions and millions of cells on in those colonies, okay? Hence why I said if your bread was moldy tomorrow, but you didn't think it was moldy today, it was, you just couldn't see it yet, okay? Those cells were present, they just hadn't multiplied enough yet. How are we doing? So far, so good? Okay. Kind of hit on some of this, all right? When they're in a colony, the cells are going to grow exponentially, but eventually they're competing for nutrients and for resources. The cells that are on the edge of the colony have little competition. They're more fit than the cells of the middle of the colony. When we put cells on a plate, we're going to use a pure culture technique that's called the streak plate method. This is by far the most common technique in all of microbiology, okay? So what's going to happen here, it allows for a dilution of cells on the plate. We're going to do this, we're going to create what's known as a 4-phase streak. So what we're going to do is we're going to take our Petri dish and we're going to divide it into basically 4 quadrants, okay? And what you'll do is you'll get the organism on your loop, you're going to streak it in one area, you'll sterilize your loop to kill everything, and then you'll be successively diluting down each quadrant thereafter. So we'll start and we're going to go back and forth along the first area, always covering new space. So you've got maybe several thousand cells on your loop to start with, and you've spread them out all along this top path. You will then sterilize your loop to kill everything that's off, and now you're going to pull a small number of cells from here farther down. So you'll go back into this, and then you'll move them all down this way. You'll sterilize, you'll come back, and then you'll take a small number of cells, and you will move them this way. You'll do it one last time. Sterilize, let it cool, and a small number of cells, and you will pull them all apart. What this is doing is taking the concentrated number of cells up here and spreading it out around the plate so that by the time you're done, you should have individually isolated colonies that you can do something with, okay? So it's a solid surface dilution that allows you to spread the cells out so that you end up with individual colonies by the time you're finished. You are going to do this once now. You will do it again after spring break. We're going to practice that technique. And then that's the first technique that's involved in the unknown experiment, which is exercise 18 in your lab manual. Okay? So we're learning about these organisms, how they're growing, what we're using them for, how we are dealing with them in class. Okay? Please understand that as a group, prokaryotes inhabit nearly all environments. When I said last week we could spend all semester trying to figure out what was on the plates that you swabbed, I meant it. Because you could have picked up nearly anything from what was on your hands or from what you swabbed. Most of these organisms like to live in comfortable habitats, but you're always going to be organisms that live in harsh environments as well. You can get organisms that live in deep sea thermal vents. Okay, you can get organisms they found live organisms living in glaciers. All right, they can live all over the place. Most that are in comfortable environments are more likely to be basic bacteria. Those that are in more harsh environments, especially when it comes to extreme temperature, are typically going to be archaea. But you've got a lot of different factors that influence growth of these organisms. Temperature, gas, pH, water availability, all of that makes a difference. We are going to regulate all of that in class. We are actually going to do different exercises in this class that look at the effects of these 4 different sets of conditions. So we'll vary the temperature and see what grows. We will vary oxygen or gas concentration. We'll vary the pH. We'll also give it a different amount of water, okay, by adding extra solute to see what happens to growth in those particular environments. All of these things concern the way microorganisms grow, and this is no different. So temperature, think about it. You want to have a hamburger, okay? You cook the meat. You understand that when you raise that temperature, you're killing the bacteria. You have leftovers, where do you put them? The fridge. You lower that temperature, that's going to, it's not going to kill the organisms, but it's going to prolong the life of whatever that food is. Same thing happens with oxygen. As you have availability of different gases, you either drive growth or you prevent growth. If you ever buy a canned food product, that canned food product is under pressure. They've removed all of the oxygen gas from that as a preservation technique. pH is another way in which we work with organisms. Your stomach kills the vast majority of the bacteria that you eat every day. Why? What does your stomach make? Acid. The stomach acid that you have kills off the vast majority of the bacteria that you eat every day. I know you want to think you never eat bacteria, and everything that you eat is totally fine, okay? But it's totally wrong, okay? You are ingesting organisms all the time. If you are going to lick your fingers and get whatever's on there off, you are eating the bacteria, okay? If you've been to the salad bar, you've touched the, spoons that are all over the place. You're coming in contact with these bacteria, they're ending up in your food. You're not constantly sick with food poisoning because your stomach acid kills lots and lots and lots of bacteria. It's coming back to this difference of pH. You buy a frozen dinner at the store. It's full of salt. It's full of salt as a preservative, right? So I know this is an exhaustive list of terms, all right? But these are terms that you already understand. You've heard of these things, okay? So when it comes to temperature, phyl on the end, all right, is referring to a condition that these organisms like. So psychro, whether it's psychrophile or psychrotroph, basically means they like it cold. A mesophile likes a moderate environment, and a thermophile likes it hot. Hyperthermophile likes it really hot. I am not, you are not, memorizing temperature ranges. You are not memorizing pH ranges, nothing like that. Our textbook has a different range than every other textbook you look at. Everything is going to be slightly different. With all of these, there's a gray area of what's considered psychrophile versus psychrotroph. I want you to think about the practical application here. If it says psychro in front of it, that means it likes it cold. Mesophile, moderate. Thermo, hot. If it is causing us to get sick, it's a mesophile because that's coming back to human body temperature, okay? Oxygen availability, you have different terms with this as well. Obligate means required. So obligate aerobe, it's required to have oxygen. Obligate anaerobe, required to have no oxygen. Facultative is kind of a wishy-washy term, basically means it can have it or not have it. So you have the term here, facultative anaerobe. These guys grow best if oxygen is present, but totally survive without it. Microaerophiles require small amounts of oxygen, but higher concentrations kill this guy off. Aerotolerant don't care at all about oxygen. So if you understand in general what you're dealing with here, you are going to be fine. The best way to get through these terms is to make yourself some flashcards, okay? If you get an exam question and you don't know what it means to be a microaerophile, you're never going to be able to answer the question. So the terminology is half the battle with this. What you see with each one of these is that you've got the categories, the classifications of the prokaryotes. Within each one of these, you have a minimum, you have a maximum, and you have an optimum range where their growth rate is best. Okay? Anything above the minimum, these guys are metabolically active. They're fine. Below the minimum, they're not doing anything. They're sitting there. They're not dead. They're just not doing anything. Anything above the maximum, those cells, their proteins have actually unraveled, and the cells are dead. Okay? But with all of these, note that there's some crossover here. Okay? There's crossover. Look at all of these guys here. There's not a whole lot of difference between those. So again, when I say there's a lot of gray areas here, it does indeed mean you can get an infection with an organism that's considered a psychotrope because it's still going to survive in your body. You take a piece of raw chicken and it's got salmonella on it. Salmonella is a really nice mesophile. You put it in the freezer, okay? You're down here, 0 degrees Celsius. That organism's not growing. It does not mean it's dead. When you thaw that chicken, the salmonella comes right back out and goes right back to multiplying again, okay? So freezing something is not killing off the bacteria. It's simply prolonging the life by lowering the temperature to a point in which that prokaryote doesn't have a metabolic rate anymore. What do we think? Oxygen. Yes, you know, you need oxygen, okay? Not all organisms use oxygen. Not all organisms that colonize your body use oxygen. You actually have anaerobes that are found in your mouth, believe it or not. Back to your gingivitis, okay? Your gums, they make a really, once you get that nice pocket between the tooth and the gum, it really does seal itself up. You get a really nice anaerobic environment that lives in there. So the importance of going back and brushing, flossing, and all of that lovely stuff is to get those organisms out of there. So you do have anaerobic organisms. Your gut is full of lots and lots of anaerobic organisms, right? So these guys can be found all over the place. You want to understand the terms here. Again, obligate aerobe means it requires oxygen. These guys also have lots and lots of enzymes that are involved in breaking down toxic oxygen radicals to make sure that this organism survives. A facultative organism also has some of these enzymes. Again, if it ends in ASE, you should know it's an enzyme, okay? Oxygen is going to be toxic to organisms unless they can break it down. And they break it down through the use of enzymes. So we classify these organisms based on how many enzymes they have and whether they're using oxygen or not. So obligate aerobes are required to have oxygen. They use it as part of their metabolism. Facultative anaerobes grow best in the presence of oxygen but can also survive without it. They have the enzymes. They can typically do aerobic respiration. They can also switch to fermentation if they need to. Obligate anaerobes are typically going to not have any enzymes. They die in the presence of oxygen. These guys are required to have an anaerobic environment all the time for their metabolism. Microaerophiles need a small amount of oxygen, typically somewhere between 2 and 10%. What's atmospheric oxygen? Concentration. Somewhere like 18 to 20%, okay? These microaerophiles are not growing on your desk. They're not growing in the toilet. They're not growing at normal environments, okay, unless we lived in Denver or something, okay? They are growing farther down in a water column or down in the soil. They're growing higher up in the mountains as well. If they do not have this 2 to 10% oxygen, they do not survive. So they have a small amount of enzymes, but they can't tolerate atmospheric oxygen. So where they grow is a very unique environment. And then lastly, you have aerotolerant organisms. These guys don't use respiration in any way. They don't care whether oxygen is there or not. Aerotolerant organisms are different from facultative anaerobes. I know the growth pattern in this broth looks similar, all right? But the difference here is that facultative anaerobes are actually using the oxygen. They can grow with it or without it. Aerotolerant organisms go, I don't care if it's around or not, it doesn't bother me, okay? But you've got to give an organism its right oxygen concentration. When we grow most plates in class, we're going to grow them in an oxygenated environment. We will try an exercise where we take away the oxygen so that you can see how that growth then changes. Water availability also makes a difference. You guys should know this. Water is the one compound in highest abundance in any living cell. We talked a little bit before about how water moves freely. What's that process called?
Osmosis.
Osmosis, okay? So water's moving by osmosis. It moves freely in and out of a cell. Water's not available in all environments. In environments with high salt, water gets driven out of cells, okay? Bacteria have to work to maintain a really nice, stable internal solute concentration. Otherwise, they can ultimately die, okay? We've covered this a little bit before. Remember, water is flowing to dilute down wherever there is more solute. So in this particular cell scenario here, you've got less solute inside, more solute outside. Water is going to be driven out of that cell, which means, so remember, in purple is the cell wall. This in green is the cytoplasmic membrane or the cell membrane. This guy is shrinking down, so this is known as plasmolysis, where this guy shrinks to a point, it might not survive anymore, okay? If you dehydrate these cells because you're pulling all the water out, this guy is not going to be able to survive. One way of doing preservation techniques is to remove all the water. So putting something in an environment with a high concentration of solute is a good preservation technique. You go and you buy a can of peaches from the store, and they're in heavy syrup. Guess what? That heavy syrup is full of sugar. You've created a high solute environment outside. So any bacteria that went into that can on those peaches, they're going to lose all of their water to that external environment. They're going to die, okay? So regulating salt concentration is a good way to control microbial growth. We play with this all the time in food production. We doing okay? A lot of this is stuff you have heard of before. A lot of this makes sense. It's things that are impacting you on a daily basis. If we're going to grow prokaryotes in the lab, not only do we have to give them those right environmental conditions with temperature and pH and gas concentration, we also have to give them the right nutritional compounds that they need as well, okay? So we have to consider what is a required element, a growth factor, their carbon source, energy source, their overall nutritional diversity. Here is part of the spot where your chemistry is going to come in, okay? You remember back to your periodic table of the elements, okay? Major elements that are required for macromolecules. There are 6 of them, 6 main ones, okay? Carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. Those are the main ones that are used. Additional ones that are major elements for bacteria include potassium, magnesium, calcium, and iron. All 4 macromolecules require high numbers of these elements. What's a macromolecule?
Carbohydrates, fats,
Proteins, amino acid, or nucleic acid, okay? So 4 main types of macromolecules make up all living organisms. Carbohydrates, so sugars, you've got lipids, which are fats, proteins, which are made of amino acids, and you've got your nucleic acids, so your DNA, your RNA, okay? Those 4 main types of macromolecules are going to contain large amounts of these major elements, okay? Organisms are then also classified, because they are organic, okay? We need carbon, based on what kind of carbon they use, okay? So heterotrophs use organic carbon as a nutrient source. Autotrophs use inorganic carbon, and when we say inorganic carbon, that means it's got to be in the form of carbon dioxide gas or CO2. Us as humans, do you think we are heterotrophs or autotrophs?
autotrophs.
We are heterotrophs. We are bringing in organic carbon, okay? You eat a salad, that lettuce is full of good organic carbon. All of the food you are bringing in is full of organic carbon, okay? So heterotrophs are using organic carbon as a nutrient source. It's carbon that is tied to hydrogen and oxygen, okay, in those compounds. If you are not able to use that organic form of carbon, you are going to be using inorganic carbon, which means it's in the form of CO2. I see a lot of blank stares. If you remember nothing about your chemistry, go back to chapter 2 of the textbook. It's not a required component for this course because it's a prerequisite, okay?
Why are we autotrophs?
We are heterotrophs.
No, you said we were both heterotrophs and autotrophs.
No, I said I was hearing both answers.
Oh.
Yes, we are heterotrophs, not autotrophs.
What
We
if
do not.
were both?
If we were both, we would have very different respiration and all kinds of other, yeah. We wouldn't have our internal structure that we need if we were autotrophs. So major elements are needed. We also have trace elements that are needed, things like cobalt, zinc, copper. Those are required in minor amounts.
I have a question.
Yeah.
What's an example of an autotroph?
Most autotrophs are actually going to be archaea in nature, things that fix carbon dioxide from the environment. Everything we're coming in contact with is a heterotroph. Okay? All these bacteria are heterotrophs. We're going to get to that in the diversity chapter. You get some organisms that fix sulfur instead. They bring in CO2 as a source. What's that?
Use sulfur as a-
They can actually use sulfur as an energy source and then fix that in the environment. Or methanogens. So
Is this like a plant or an organism?
it's an organism. They're typically they all fall under archaea. So like methanogens end up, they bring in certain compounds, they use hydrogen, gas, other compounds, and then they basically release methane gas. So methanogens releasing this methane gas are found in cow guts, yeah. So we have all this biofuel where they collect the,
That's where most methane comes from.
that's where they get all this methane from, and all these farmers, they basically take, why am I not capable of talking today?
I know where you're getting from.
They're basically, they take everything from the pasture and they put it in these pits, and they put tarps over the top to collect the methane gas. The methane gas is actually coming from the methanogens, which are living in the gut of the cow. Biofuel. But they're archaea in nature. This is no different than you taking a multivitamin. If you've ever looked at a bottle on a multivitamin, it has a list of all the large numbers of nutrients that are at the top, and it has a whole list of all these ones that are in teeny tiny amounts at the bottom, okay? We are no different. We have some things that we need in large amounts, like carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. And then we have a whole bunch of things that are useful for how our enzymes function and making our body work properly, but we only need them in small amounts, okay? Bacteria are very similar to us in terms of their nutritional requirements. You're not memorizing the table, okay? I'm putting this up here to help you see all of the ways in which these particular elements are necessary. So carbon, oxygen, and hydrogen, these three are basically making up, quote, organic compounds, okay? You need this in all kinds of cellular components. Nitrogen is needed in large amounts to make up amino acids. What do amino acids get put together to make? Proteins, okay? Nucleic acids are your DNA and RNA. They require nitrogen. So if a bacterial cell is going to divide, it has to copy its DNA. If it doesn't have a good amount of nitrogen, it can't make more DNA because it's missing an important component of that particular macromolecule, okay? Growth factors are additional compounds that some organisms need that they cannot make on their own. They have to be provided to it. Now this is going to be different for every species, For example, E. coli doesn't require any growth factors. It can actually make everything it needs. E. coli is one of the easiest organisms to grow, believe it or not. It's not really finicky when it comes to what it needs. However, other organisms need growth factors that have to be added from the environment, otherwise they're not capable of surviving, okay? For example, there is a growth factor for humans. It is required for a developing fetus to help make sure the spinal column fuses and that your brain and your spinal column are properly formed. Does anybody know what that growth factor is? Nope.
Folic acid?
Folic acid, okay? Folic acid is that one compound that gets added to all kinds of prenatal vitamins. The difference between a regular vitamin and a vitamin for a pregnant woman is folic acid. You need large amounts of folic acid if you are growing another human being because it's required for its proper growth and development. Folic acid is fortified into all kinds of products. Go home and pick up a cereal box. It will tell you how much folic acid has been added, okay? We do not care about how much folic acid you have unless you are growing another human being. Gentlemen, you never have to care about how much folic acid you are putting into your body. Doesn't matter. Unless one day you are growing another human being, maybe that is possible.
Who knows?
Then you have, who knows, okay?
Very interesting.
That is true, okay? But it's a growth factor for us. We cannot make it. We have to get it from our external environment, okay? Bacteria are the same way, but it's going to be different for the species. If we want to grow a certain species in the lab, we need to understand what its growth factors are, if it has any. Okay? So we covered carbon source, all right? We talked about that autotroph versus heterotroph. Also understand organisms have to have an energy source. Energy in the cell is in the form of what?
Glucose.
ATP. Okay? Energy in a living organism has to be in the form of ATP. Okay? If a cell gets their energy from the sun, it takes that sunlight and it converts it into ATP. It is called a phototroph. Organisms that get their energy from chemical compounds are called chemotrophs. Us as humans, are we phototrophs or chemotrophs? We are chemotrophs, okay? It just so happens that all that food that you are eating in while you are in here is not only your energy source, but it also functions as your carbon source as well, okay? We typically group them according to energy source and carbon source together, okay? So these terms get combined. So if you see photo in front, it means your energy source is from the sun. If it's a photoautotroph, it means it's energy from the sun, but its carbon source is in the form of CO2. If it's a photoheterotroph, it means its energy is from the sun, but its carbon is in the form of organic compounds. When we get into the chemotrophs, we get a couple of additional terms. So chemolithoautotroph. If you see chemolithoautotroph, it means everything is inorganic. So it means it's using an inorganic chemical as its energy source, and that means it's using inorganic carbon as its carbon source. Okay? If you see the word chemoorganoheterotroph, it's organic for the energy and organic for the carbon source. You will not always see that organo in there. You may just see chemoheterotroph, because all of these heterotrophs need organic carbon, okay? So if it just says chemoheterotroph, it's organic energy, organic carbon.
You said we are chemo-organoheterotroph?
Right, we are chemoorganoheterotrophs. Almost all of the bacteria you will ever encounter are all going to be chemoorganoheterotrophs. They're feeding off of the same things that you like to have, okay? All organisms need a carbon source. All organisms need an energy source. You need the energy source because that's how you make the ATP, and the ATP is needed for cellular division and growth. You need the carbon source because the carbon source is needed for cell growth and division as well, okay? We have got to make sure if we're going to grow microorganisms, we give them an energy source, we give them a carbon source. The media we use in lab is full of sugar. It's a basic way to feed these guys and give them both a carbon source and an energy source, okay? We're going to grow things in the lab. We have to keep track of what we're growing and how we're growing it. We can vary the different types of media to do this, okay? And we have all kinds of different classifications of media. I don't want you to memorize different types of media here. We're going to get more into that in the lab, all right? But what I want you to understand is if we don't give an organism what it needs, it dies. Yes, there is a media called chocolate agar. Okay?
Made of chocolate.
It's got chocolate in it. There are certain organisms that really just prefer to grow on that chocolate agar. Okay? We grow blood agar. We use blood agar a lot. Blood agar is going to grow pretty much everything, but blood agar is actually going to be differentiating certain organisms based on their hemolytic properties. If you go in, you think you've got strep throat, they can do a rapid test. If your rapid test is negative, they'll say, we can send this out for culture if you like. What does that mean? That means they're swabbing your throat and they're putting it on a blood agar plate. In 24 to 48 hours, if you see these clear areas in the blood agar, it means you are positive for the organism that causes strep throat. It's the only one that grows in that environment that causes that, okay? So by varying the different types of media, we can vary what grows. We can vary the output as well. Different types of media are classified differently. So we can have complex media versus chemically defined media. Complex media is just a variety of ingredients. There's no exact formula versus defined media where everything is precisely weighed. Some organisms are very finicky and they need everything in exact quantities versus other organisms need things that are just kind of in the right ballpark. This is no different than cooking versus baking. If you're cooking and you don't like the taste of something or you want to go easy on the pepper or you're watching your salt intake, you just vary all of that. But if you're baking, you better have the baking powder, baking soda, all of that in the exact right amount or you're not going to get the leavening that you need of your baked goods, okay? Or this is the difference between a patient who can get a regular hospital lunch versus a patient who's on a feeding tube, okay? Everything is precisely controlled. Different types of organisms require different amounts of nutrients. We are going to use different types of selective and differential media in class. So selective media inhibit the growth of unwanted organisms, and they only allow other things to grow. So for example, Thayer-Martin agar only allows Neisseria gonorrhea to grow. If you work in an STD clinic, you have Thayer-Martin agar available. What do? You take a swab from that patient and you streak it on this plate. If there is any growth whatsoever on there, it's got to be that patient has gonorrhea. Okay, that's the only organism that grows on that plate due to the selective agents that are in there. Okay, we will use McConkey's agar in class. It only grows gram-negative bacteria. We've added in additional compounds that prevent the growth of everything else from growing on there. So it's a way of selecting what we want. We can also have differential types of media that allow you to differentiate what's on the plate. So this is going back to that blood agar. So do you see how this blood agar is kind of going orange here? You're getting these clearing areas around it. That's what's going to happen if you have strep throat with a throat culture on a blood agar plate. Okay? Most other organisms will grow. A lot of them end up turning the blood agar green. But when we see this clearing, that's a sign of hemolytic properties. The only organism that can be possible in your throat with hemolytic properties is going to be Streptococcus pyogenes, which is causing your strep throat. Okay? So lots of different types of media. We can vary what's in there to help us understand what's growing or to control what's growing. We can alter gases in the laboratory as well. What you're looking at here is a gas chamber. We are going to use this in class. So what we do, we put the plates in here. We put what's called a gas pack. The gas pack actually releases hydrogen gas. That hydrogen gas is actually going to react in here. It's going to create condensation, so it'll create water that ends up on the wall. Everything else, once we've gotten rid of that oxygen, this will be an oxygen-free environment. It's got a one-way gasket that allows extra gas out, and it basically creates an anaerobic environment inside of here. You can have specialized incubators as well, culturing facilities that will allow you to basically put your hands in, but they're totally anaerobic. So if we wanted to grow organisms that do require a no oxygen environment, we can actually find ways to do that. We can detect bacterial growth in all kinds of different ways. No, you're not memorizing this, okay? The thing I want you to understand is that we do this all the time, and you've seen this all the time. A big way that we're going to look at growth of microorganisms in class is to look at turbidity or cloudiness. If you've ever known a patient who's been on a catheter, okay, what's coming out of that catheter should be clear. If it's cloudy, you see that bag and it's cloudy looking, what do you think? UTI. They've got a urinary tract infection. Because what should be coming out of there should be sterile. If you ever look at a patient who's got a catheter in and that urine bag is cloudy, you need to say they've got an infection there. That's what's going on, okay? You pick up a juice from your car that's been sitting on the floor, Gatorade, okay? And you see it's all cloudy. What do you do with it? You throw it away. You see the gallon of milk, and the gallon of milk is expanding. All right, have you ever crushed a gallon of milk, empty gallon of milk, and then you come back later and you're like, why is it expanding on me? It's producing gas. There are bacteria in that gallon of milk that are producing gas. You want to know if your gallon of milk is good or not, what do?
Smell it.
You smell it. Because those organisms that are in there are going to be giving off acid, and you know when your milk is not smelling right anymore. It's smelling a little sour, okay? So we do this all the time on a regular basis. Yes, we can get a little bit more fancy. We can do direct counts. We can put it under a microscope and count how many cells that are there. There are lots of different ways of actually detecting bacterial growth, and you do this on a regular basis. Again, something that does not have a lot of cells in it should be more clear. Something that's cloudy has a large amount of cells. We have ways of basically counting this where you shine a light through it. The amount of light that passes through to a meter actually tells you what percentage of cells are in there. Okay, so you can visually look at something and say, okay, I know this has more cells than this one. You can also pass that through a counter to detect that in the end. Okay? All right, we have lots of different ways of detecting those products, whether it be acid, gas, ATP production. There's a number of different ways in which you can do that, okay? What I want you to do now, we've got a few minutes left. I don't know what that clock says, but I'm going by my watch, okay? I'm passing out a sheet of paper. I want you to take one and put your name on it, please. Close your notes. Oh boy. I'm sorry. I'm really not, but. All right, so in this class, you get participation points for showing up to class. So what I want you to do today is to draw a coccus-shaped bacterial cell. I want you to put what organelles or structural components it's required to have, and I want you to label them. And I want that to include its external parts as well. So, you are drawing on your piece of paper a coccus-shaped, so get your shapes right, bacterial cell. You need to label its parts. You need to have whatever required components have to be there. So what do all prokaryotic bacterial cells have to have? And I'm going to tell you it's a gram-negative cell. Because that is what's going to help you with your external components. You can talk talk to the person next to you. Don't open your notes. It's gotta be a gram-negative, coccus-shaped cell.
Yeah, I have no idea.
Talk to the person next to you. We spent 2 lectures going through this material. If it's coccus shape, what's its shape?
Circle.
Circle. Okay? If it's gram-negative, that helps you understand what its external cell components look like. Like what's in the cell wall? What does it have? What does it not have? Prokaryote.
Allow the dye to go into it so it can.
What time are they done with lab? I left my jacket in there.
There is nobody in there now, but I'm going back in there at 1:30. It's locked. I locked it when I left.
Okay. It's like, why am I so
Circle kind of
cold?
looks like an egg.
You want us to like label literally everything inside of the cell that should be inside of the cell?
That it has to have.
Oh.
Give me what it has to have. It's prokaryotic, so your numbers of inside components should be fairly limited.